Hello students, this is molecular basis of inheritance part 8. In the earlier slide we have seen that how a leading strand get formed. Now this is a leading strand, there is DNA polymerase which is moving forward to the replication fork. Now this is another strand that is lagging strand. See, there is 3' to 5' and this is Okazaki fragment, and that blue colored segment is the RNA primer. Now see how the primase forms the primer and the DNA polymerase enters DNA bases. They get added to 5' to 3' end, from 5' to 3' end. Now again there is attachment of primase. Again DNA polymerase gets attached. Again the activated nucleotides, they get added, and there is formation of a strand. Now another enzyme, now this is RNA primer. Exonucleases, they come, they remove the RNA primers, and again the DNA polymerases, they work. Okay, so this is how the primases, they are eliminated, and one strand gets formed in continuation manner, that is leading strand, and another DNA strand, it's get formed in the discontinuation manner. Now this is the old strand and another is the newly formed or the daughter strand, and the DNA replication works in this manner. So what is leading and lagging strand? Now let's try to understand it. The template strand with the free 3' end is called as leading template. Here we are talking about the leading template. Template is the old strand. Okay, so the old of that old strand of the DNA, here we are talking about the free 3' end. Okay, so the template strand with the free 5' end is called as lagging template. Leading template has 3' end, lagging template has 5' end. This is the basic difference between the leading template and the lagging template that you have to remember. Okay, so both the strands of the parental DNA, they are anti-parallel. New strands, they are always formed in the 5' to 3' direction. Why the Okazaki fragments they are required, or why both the strands cannot get formed in continuous manner, why both the strands are not leading strand, but on the contrary, one is leading strand and another is lagging strand? Because we all know, when you're talking about the template strand, or when you're talking about the old double helical structure of the DNA, so in that DNA structure we have studied that DNA is anti-parallel. Okay, two strands, they are anti-parallel. So because of that, one is with 3' to 5' end, another is 5' to 3' end, and new strands, they are always formed in 5' to 3' end. So one with 5' to 3' end, it can get formed continuously, but another with 3' to 5' end, there are complications. So for that, the strand activity is it is little complicated, and it occurs in the segments or small chunks of the DNA segments. Now in this slide we are going to discuss that how the maturation of Okazaki fragments occurs. So a DNA synthesis on lagging template takes place in the form of small fragments or small chunks, is called as Okazaki fragments. Now you can observe in this diagram, this is the leading strand from 5' to 3' end, as the direction is 5 to 3'. Okay, so this occurs in the continuous or in the form of leading strand. Another is lagging strand. Okay, now see the lagging strand, 5 to 3, again 5 to 3, again 5 to 3. In that segments, the Okazaki fragments, they get formed. Why there is discontinuation? Because there is 3' to 5' end. Can you observe that 3' to 5'? Okay, so the DNA polymerases, they can act only from 5' to 3' direction. So we need the 5' end, and that 5' end is provided by the RNA primer. Now that dotted segment is nothing but the RNA primer. It provides the attachment for the DNA polymerase. So at this point, the DNA polymerase gets attached and it gets moved to the next blank space, to the next RNA primer. So this arrow is the lagging strand of the DNA, and again the DNA primer, RNA primer, and again there is formation of a strand. So it occurs in the segments, okay, and that's why this strand is lagging strand and it is discontinuous. Remember, it is because of the 5' to 3' direction of the strand, and both the strands, they cannot have 5' to 3' directions or two ends same, because they are anti-parallel. Okay, they run oppositely. One is with 5' to 3', another with 3' to 5'. Okay, that you have to remember. So the DNA synthesis on lagging template takes place in the form of small fragments or small chunks. These are known as Okazaki fragments. Okazaki is the name of the scientist. So these fragments are joined by an enzyme that is called as DNA ligase. Okay, so the RNA primers, they are removed. After the removal of the RNA primer, there will be empty space. So these fragments, again, that empty space, it is joined by this DNA ligase. DNA ligase is that name you have to remember while talking about Okazaki fragments and their maturation. So the RNA primers, they are removed by the enzyme RNAase. RNA primase, they eat or they cut or they dissolve or they help in the disappearance or disappearance of this RNA primer. Okay, they eat. In short, the RNA primase, it eats the RNA primer segment, or it removes the RNA primer segment. Finally, the DNA gyrases. These are also called as topoisomerases. These enzymes, they form double helix to form daughter DNA molecule. Now let's talk about what are topoisomerases. So these topoisomerases, or the DNA topoisomerases, these are the enzymes that participate in the overwinding or underwinding of the DNA. So talking about it, underwinding or overwinding of the DNA, these topoisomerases, they play very important role. The unwinding problem of the DNA arises due to the intertwined nature of the double helical structure. During the DNA replication and transcription, the DNA becomes overwound, or the replication forks as it moves forward. So there is some kind of tension or strain, it occurs. So if left unabated, this torsion or the force or the tension, it is called as torsion, would eventually stop the ability of DNA or RNA polymerases involved in the process to continue down the DNA strand. The replication process, it cannot go that smoothly because of the because of that tension or because of the torsion which is occurring because of more twisting of the DNA. So to release that strain, the topoisomerases, to release that torsion, these topoisomerases or DNA gyrases are very important. Because of these group of enzymes, the DNA replications, it occurs very smoothly, very continuously, without any disturbance. There is one animation you can observe. In this animation, how the double helical structure get untwisted, and there are a number of enzymes, they are working together. Now these are the number of enzymes, they are working together. This is leading strand and there is a loop formation. Okay, can you observe that loop? See, this is the loop formation and these are the Okazaki fragments. Okay, so there is a loop formation. This is the lagging strand. So the newly synthesized strand develops continuously towards the replication fork. Now see, there is a replication fork. This is the replication fork. This is twisted DNA structure. Can you see that? This is the twisted structure, and these enzymes, they are moving forward. This is helicase enzyme which is moving forward. This is separating these two strands. So there is 5' to 3' direction of the strand. This is DNA polymerase which is working in a continuous manner. It is adding the nucleotides and the twisted strand is getting formed. So out of these two twisted strands, one is newly formed, okay, and another is old strand. Now see, this is the old strand, okay. And talking about this lagging strand, the strand which develops discontinuously away from the fork. Now this is away from the fork. This is replication fork, and the synthesis of the strand is away. Okay, so it forms a loop-like structure. Okay, and there is formation of a loop, and there is formation of there is addition of the nucleotides, because the 5' to 3' end, that is the basic requirement for the DNA polymerase for its activity. Okay, so this is the beautiful animation which you can observe. So I hope it is very much clear that how the DNA replication it occurs, and what are the leading and lagging strands, and how it occurs inside our cell. So this is the end of replication process. This is the last step of DNA replication, that the formation of daughter DNA molecules. So you can observe in this diagram, this is double-stranded DNA. These are activated nucleotides. They are getting added. One is leading strand, another is lagging strand. Pink color is the metallic color is the old strand, and the blue colored strand is the daughter strand or newly formed strand. So at the end of the replication, we get two daughter DNA molecules on the template, old template DNA strand. Each daughter strand, DNA strand, one is parental and other is newly synthesized. One is parental DNA, another is daughter DNA. It is described as semi-conservative replication. Semi means half. Out of these two newly formed DNA, out of these two strands, one is old strand while another is newly formed strand. So one is the parental DNA, another is the daughter DNA. One is parent DNA, old strand, another is daughter strand. So because of that, it is called as semi-conservative replication. So I hope your all concepts are very clear now, and it is very important. DNA replication, this process can be asked. The number of enzymes which are involved in this process are very important. So please do try to understand all these steps. Remember all the name of the enzymes, remember their functions, and there are no complicated matters which I have discussed. It is very beautiful phenomenon which occurs, which is now occurring in your body. That is the beauty of science, that is the beauty of biology. So that's all of formation of daughter DNA molecule. So that's all for about the DNA replication process. Keep your textbook with you while studying this process. It is very beautiful phenomenon, and you'll be able to understand it if you read the matter once, twice, thrice, and keep learning this video, as I have added number of animations in it, and I have used different colors for different terms. For enzymes, I have specified the name of enzymes with the pink color only. This is again very important. If you are preparing notes, do try it, and it will be very easy to remember the name of the enzyme and the name of the enzyme, function of the enzyme. So if you are going to study in that way, the DNA replication becomes very easy. It is very important if you're talking about genetics, and if you're talking about DNA replication, to understand the structure of DNA, to understand the replication of DNA, because we are going to study the protein synthesis and the operon concept, which is again very much complicated. So let's not make it much complicated. Try to observe the beauty of cell, try to observe the beauty of science, the beauty of biology. So that's all for today's session. Thank you.